Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Now, scientists at Scripps Research have uncovered new details about the immune system's role in this cycle. They reported in the journal Brain, Behavior and Immunity on February 28, 2023, that the immune signaling molecule interleukin-1 beta, IL-1 beta, is present at higher levels in the brains of mice with alcohol dependence. In addition, the IL-1 beta pathway takes on a different role in these animals, causing inflammation in critical areas of the brain known to be involved in decision-making. These inflammatory changes to the brain could explain some of the risky decision-making and impulsivity we see in people with alcohol use disorder, says senior author Marissa Roberto, Ph.D., the Schimmel Family Chair of Molecular Medicine and a professor of neuroscience at Scripps Research. In addition, our findings are incredibly exciting because they suggest a potential way to treat alcohol use disorder with existing anti-inflammatory drugs targeting the IL-1 beta pathway. AUD is characterized by uncontrolled and compulsive drinking, and it encompasses a range of conditions including. Researchers have previously discovered numerous links between the immune system and AUD, many of them centered around IL-1 beta. People with certain mutations in the gene that codes for the IL-1 beta molecule, for instance, are more prone to developing AUD. In addition, autopsies of people who had AUD have found higher levels of IL-1 beta in the brain. We suspected that IL-1 beta was playing a role in AUD, but the exact mechanisms in the brain have been unclear, says first author Florence Varadayan, Ph.D., an assistant professor at Binghamton University and former postdoctoral fellow in the Roberto Lab. In the new study, Roberto, Varadayan and their colleagues compared alcohol-dependent mice with animals drinking moderate or no alcohol at all. They discovered that the alcohol-dependent group had about twice as much IL-1 beta in the medial prefrontal cortex, MPFC, a part of the brain that plays a role in regulating emotions and behaviors. The team then went on to show that IL-1 beta signaling in the alcohol-dependent group was not only increased, but also fundamentally different. In mice that had not been exposed to alcohol, as well as in mice that had drunk moderate amounts of alcohol, IL-1 beta activated an anti-inflammatory signaling pathway. In turn, this lowered levels of the inhibitory neurotransmitter gamma-aminobutyric acid, GABA, a signaling molecule known to regulate neural activity in the brain. However, in alcohol-dependent mice, IL-1 beta instead activated pro-inflammatory signaling and boosted levels of GABA, likely contributing to some of the changes in brain activity associated with AUD. Notably, these changes in IL-1 beta signaling in the alcohol-dependent mice persisted even during alcohol withdrawal. Drugs that block the activity of IL-1 beta are already approved by the U.S. Food and Drug Administration to treat rheumatoid arthritis and other inflammatory conditions. More work is needed to determine whether these existing drugs could have utility in treating AUD. We plan to follow up on this study with more work on exactly how targeting specific components of the IL-1 beta pathway might be useful in treating alcohol use disorder, says Roberto. Thanks for watching. Please subscribe my channel for more videos.